And we're live. Welcome, everyone. It's my pleasure to chair this new session, which promises to be yet another great session and very exciting. We will start immediately. So we are more than 800 people already. So that's great. So we will start immediately with the first talk by Yushu Chen. Hi, Yushu.
Hi.
How's it going?
Good.
Yushu is from Cold Spring Harbor Laboratory, and today you're going to tell us about the wiring logic of the rodent olfactory system revealed by sequencing of single-cell projection. Stage is yours.
Okay, let me share my screen. Do you see my screen okay?
Yes, absolutely.
Okay, first I'd like to thank the organizers for giving me the opportunity to present my work at the Cosyne conference. I will talk about the neuronal connections in the early rodent olfactory system. Neuronal computation relies on the structure of intra and interbrain region connectivity. Determining the logic of a neuronal connection is therefore critical for understanding principles underlying neural processing. The connections from sensory periphery to cortical areas are spatially organized in the somatosensory, visual, and auditory system. The spatial mapping of sensory properties thus appear to be a general strategy that is a nervous system use. However, whether a similar spatial organization of sensory information exists in the olfactory system is a matter of debate. In the rodent olfactory system, the olfactory bulb conveys the odor information detected by sensory neurons in the nose to the rest of the brain. Sensory neurons that express the same receptors converge on the same glomeruli on the olfactory bulb surface, and the glomeruli activation is routed by the output neurons called muscle and taft cells to higher brain regions. Muscle cells, as shown in blue and red here, project broadly to all these major target brain regions. Taft cell in green here project sparsely to the anterior part of the olfactory system. So in this way, the odor information is broadcast across the brain. Our question is there structure in olfactory bulb connectivity to higher brain regions as in other sensory systems? Previous anatomical studies based on the bulk tracing, retrograde tracing, and single-cell tracing of 20-30 muscle taft cells across the brain propose that olfactory bulb projections to piriform cortex, which is the largest olfactory cortex, are highly distributed and largely random. Current computational models of olfactory processing rely on the distributed and random connectivity. It's in striking contrast with the spatially organized representation in other sensory modalities. So are olfactory bulb outputs really random or structured in other ways? So although previous studies didn't reveal the structured sensory maps in the olfactory system, these studies couldn't tell us whether any other kind of spatial organization exists in the olfactory system due to the limitation of previous studies, such as the bulk tracing like single-cell resolution, and single-cell tracing only reconstructed a very small number of cells. So we set out to re-examine the questions of the olfactory bulb projections by taking advantage of a high-throughput strategy that allow us to map the projection of many cells at a single-cell resolution in a single brain. First, I will talk about how we use the high-throughput strategy to map the projection of thousands of olfactory bulb projections. And our data reveals that multi-cell projections along piriform AP-axis are related to co-projections to extra-piriform cortex areas. We next investigated piriform outputs and found that piriform output differentially from its AP-axis. More interestingly, the spatial patterns of multi-cell outputs and piriform outputs match and form the matched input-output circuit motif. So our finding outlines the general principle of wiring neural circuits across brain that has been observed in various systems, such as visual and limbic system. So we all know the neuronal process intertwine extensively in the brain. It prohibits multiplex tracing by microscope. Only a very small number of cells can be labeled and traced by microscope in a single brain. So it's very low throughput. To overcome this limitation, we instead use short random RNA sequences, which we call the barcodes, to label individual neurons in the brain and sequence the barcodes by the high-throughput sequencing. This technology is called MapSeq. It's developed by Zador lab at the Cosyne Laboratory. Because barcodes could have enormous diverse sequences, this technology enables us to track the projection of thousands of cells at a single cell resolution in a single brain. Barcodes are introduced into individual neurons by viral infection. Each neuron is labeled by a unique barcode sequence. Within a cell, the barcodes can replicate and travel from soma all the way to axon terminals at the target brain regions. So let's look at this cell, labeled by the barcode 37. We can see this cell can project to all these different target brain regions. We can read out this cell's projection by sequencing barcodes at both the target brain regions and also within the olfactory bulb. This panel shows a projection of cell 37 at the brain area level. On the y-axis is the relative projection strength. On the x-axis are the target brain regions. We can see cell 37 project broadly to all these target brain region with the strongest projection to piriform cortex. With MapSeq, we have mapped the brain-wide projection patterns of 4,800 output neurons at a single cell resolution from 6 brains by MapSeq. It's a huge data set compared to previous single cell studies. And we can separate these output neurons into different cell types, such as multiple versus taft cells, by matching their brain-wide projections to soma positions in the bulb. Because over 90% of our data are multiple cells, so we focus our analysis on multiple cells only. This panel shows a projection pattern of putative multiple cells. The spatial resolution for our projection data is 200 micron from anterior to posterior within each target brain region. So each row represents a single multiple cell, and each column represents the projection to a 200 micron corona section within each target brain region. And different corona sections within the same brain region are arranged from anterior to posterior. The multiple cells are sorted based on the position of their peak projection within piriform from anterior to posterior. So we can see multiple cell projection basically tile the entire piriform along its AP axis. Some multiple cells have the peak projection to the anterior piriform cortex, and these cells have very little projection to posterior piriform, cortical amygdala, and entorhinal cortex. Some multiple cells have the peak projection at the middle to posterior part of piriform, and these cells have stronger projection to cortical amygdala and entorhinal cortex. So it looks like the multiple cell projections to different AP positions within piriform cortex are correlated with projection to non-piriform regions. So we plotted the relationship of multiple cell projection to extra piriform regions and multiple cell projection along piriform AP axis. On the y-axis is the mean projection strength to each non-piriform brain regions. On the x-axis are the positions of multiple cells dominant projection along piriform AP axis. So if there's no relationship at all, we expect to see a near flat line, just like here. But instead, we see a slope for multiple cell projection to LN, and multiple cell projection to LN are stronger for muscle cells that project dominantly to the anterior part piriform, then muscle cells that project dominantly to the posterior part piriform. So basically, it indicates that if a muscle cell projects strongly to anterior piriform, it also projects strongly to LN. So we see the reverse slope for muscle cell projection to cortical amygdala and entorhinal cortex. So basically, muscle cells projecting strength to cortical amygdala and entorhinal cortex are highly correlated with stronger projection to posterior piriform cortex. We also look at individual muscle cells projection to olfactotubercle, but did not see such a robust relationship. So to summarize this first part, using MapSeq, we have mapped the projection of over 4,800 output neurons from the olfactory bulb, and muscle cell projection along the AP-axis of piriform and co-projection to extrapiriform regions are highly correlated, such as muscle cell that projects strongly to anterior piriform also projects strongly to LN, and muscle cells that project strongly to posterior piriform also have stronger projection to cortical amygdala and entorhinal cortex. So our results suggest olfactory bulb projection to piriform cortex are spatially structured. It challenges the model of total random connectivity between olfactory bulb and piriform cortex. So since muscle cell projection to extrapiriform regions are correlated with muscle cell projection along piriform AP-axis, so we wonder whether piriform outputs also display a similar spatial gradient along the AP-axis. To test this possibility, we have mapped the projection of 30,000 individual piriform neurons from a fine brain by MapSeq. The left panel shows the projection patterns of these 30,000 piriform neurons. Each row represents a single piriform neuron, and each column represents the projection to a piriform target brain region. The piriform neurons are sorted based on their soma position within piriform from anterior to posterior. So we can see their soma position along piriform AP-axis on the right panel here. From this projection matrix, we can see that neurons from anterior part of piriform have stronger projection to olfactory bulb and LN than neurons from posterior part of piriform. In contrast, neurons from posterior part of piriform have stronger projection to cortical amygdala and entorhinal cortex. So basically, our results suggest that piriform cortex outputs vary with their soma locations along the AP-axis in the way they project to specific sets of target brain regions. For example, anterior part of piriform projects strongly to LN and olfactory bulb. Posterior part of piriform have stronger projection to cortical amygdala and entorhinal cortex. So the spatial pattern of piriform outputs is reminiscent of the spatial pattern of olfactory bulb outputs. So both olfactory bulb outputs and piriform outputs are spatially organized along piriform AP-axis in terms of their projection to extrapiriform regions. It looks like olfactory bulb outputs match piriform outputs. Indeed, for a given AP position within piriform, the piriform outputs to extrapiriform regions match the muscle cell outputs to extrapiriform regions and form the matched input-output circular motif. So now let's look at the first look at both piriform cortex and muscle cell projection to LN. In this plot, different AP positions within piriform are color-coded from the blue to purple. Let's first look at this blue dot. On the y-axis, it represents neurons from anterior part of piriform, and these neurons have stronger projection to LN. And on the x-axis, this blue dot indicate muscle cells that project dominantly to anterior part of piriform, also send a stronger projection to LN. Let's look at a purple dot. On the y-axis, this purple dot indicates neurons from posterior part of piriform send a weaker projection to LN. On the x-axis, this purple dot indicates mother cells that project dominantly to posterior part of piriform have weaker projection to LN. So basically, this plot tells us that for mother cells that project strongly to anterior piriform also project strongly to LN, and anterior piriform also have stronger projection to LN. We also look at piriform cortex and mother cells projection to cortical amygdala and entorhinal cortex. We also see the correlation, but just with the reverse direction. Mother cells that project strongly to posterior piriform also project strongly to cortical amygdala and entorhinal cortex. And in turn, posterior piriform have stronger projection to cortical amygdala and entorhinal cortex. So in conclusion, our results suggest a matched input-output circuit motif in the olfactory system. In one circuit, the olfactory bulb projects strongly to both anterior piriform and also LN. The anterior piriform have stronger projection to LN. In the other circuit, the olfactory bulb projects strongly to both posterior piriform, cortical amygdala, and entorhinal cortex. And the posterior piriform have stronger projection to cortical amygdala and entorhinal cortex. So these extrapiriform regions, such as LN, can receive both direct input from the olfactory bulb and also the same information after being processed by the piriform cortex, and can integrate the parallel streams of information. So it indicates the presence of structured connectivity in the olfactory system, and the matched input-output circuit motif are similar to neural circuits in other sensory systems, such as in visual and limbic systems. And these downstream brain areas can receive both direct input from upstream areas and also the same information after being processed by other brain region. So in terms of the connectivity rules, the olfactory system may be quite similar to other sensory systems. Overall, the matched input-output circuit motif is a general principle to wire neural circuits across brain, and such architecture enables different computation on sensory inputs and can integrate parallel streams of information. So this work is spearheaded by me and Xiaoyin. We also get a lot of support from Batu and other people in the Albeanu, Koulakov, and Zador's lab. And this research was supported by NIH Transformative R1 and bring R1 to the Albeanu lab and Koulakov lab. So thank you for your attention. I'd like to take questions.
Thank you, Yushu. Fantastic data. Beautiful. Everyone, just a reminder to use Slido if you have any question, and the question will be voted. So we have already actually quite a few questions. Perhaps just a time for two quick questions. So the first by Claire Messner-Bernard. Are the mitral cells projecting to the anterior piriform cortex and the one projecting to the posterior parietal piriform cortex intermingled in the olfactory bulb?
So are the muscle cells that project
within the olfactory bulb, actually, are they coming from the same location? Have you checked this?
Oh, this way. So the location within the olfactory bulb, we only have the 200 micron resolution from anterior to posterior, and we see the we don't see the segregation of these muscle cells, but maybe we didn't have enough spatial resolution.
Sure, thanks. Second question by Sophie Bagour. Did all OB mitral cells have their strongest projection to the piriform, or did any projection most strongly or exclusively to other olfactory areas?
I'm sorry, can you repeat this?
Okay. So do all olfactory bulb neutral cells have their strongest projection to the piriform, or did any project more strongly or exclusively to other olfactory areas? So is it all mostly to the piriform cortex, or do you have direct connection from the olfactory bulb to other olfactory areas?
The major cell projection is quite broad. It projects broadly to LN, olfactotubercle, and other brain regions. And the strongest projection is within piriform. From this perspective, our data are consistent with previous studies.
So do you think that there's another level of organization then directly from the olfactory bulb to the other non-piriform cortex projection?
You see, do we see whether one neuron projects specifically to one target brain regions? Do you mean the exclusive projection? We do not see much of that in our data set. And the previous single cell tracing also suggests multi cell projection is indeed broad.
Okay, I see. Thanks. So perhaps one last question from Peng Hao Sean Tan. Can you speculate on what kind of different information is being processed by the two different streams, anterior and posterior piriform cortex?
Anterior and posterior. So we only have some, we do not know the exact answer for sure. We only have some speculations. Maybe the anterior and posterior part can process, sensory and contextual information.
I see. Okay, and one last one, perhaps, I don't want to get too late. So from Cameron Harris, within OB anterior piriform cortex or OB posterior piriform cortex subcircuits, do your data show any existence of connectivity structure, which is where most theories of randomness were suggested? So can it be locally random, basically?
What do you mean be locally
random? Locally random, like from the OB to the anterior piriform cortex, perhaps within the circuit, the projections are random. Do you have any hint of that or not?
So OB projection to piriform, to anterior piriform.
Yes.
Are random. I'm not sure whether I clearly understand this question.
So you started in your talk. You started saying that actually one theory is that it's totally random, and you say no, there is structure here. There's a wiring diagram. However, locally within the two subcircuits that you have described, it can be locally random.
I see. I got that. Maybe within the anterior circuit, posterior circuit, could be. No, we do not think so. I'm not sure whether I can still go back to some of my slides.
Yeah, very quickly, but go ahead. No, can you or not?
Did you see this? Did you see we see a continuous slope within anterior part and also posterior part?
Okay.
So we speculate, there's structure. No.
Okay. Thank you again, Yushu.
Thank you.
So we're gonna actually now go to the second talk of this session, and
it's time for me to leave now.
Yes, please. Thank you. Bye.
Thank you.
So the second talk will be by Kevin Balding from Boston University. Kevin, are you here? Can you hear me?
Yes.
Hey, how you doing? And today you're gonna present us your work on bilateral sensory signals for odor source localization in freely moving mice. Quite exciting. So stage is yours, Kevin.
Okay, I'm working on sharing my screen.
It was working 20 minutes ago.
I agree. Oh my gosh. I'm doing the same process that I did previous, but maybe.
But don't worry, take your time.
The difference is that my Chrome, there we are. Have you got it now?
It seems it's working. Yes, great.
All right.
Go ahead.
Okay, so I also want to thank the Cosyne organizers for making this meeting possible, and especially to thank Leslie for her work keeping us organized and making sure we're ready for this session. Today I'm going to describe some of the work we've been carrying out in Ian Davison's lab at Boston University, aimed at understanding the types of information available to an animal attempting to navigate to an odor source, and how animals could make use of that information to guide their search. This work is very much at an intermediate stage in its development, and the data have the potential to speak to a number of related computational questions about sensory-guided navigation. So I'm looking forward to comments from this audience here and later offline. So identifying and localizing odor sources is a primary and critical function of the main olfactory system, enabling animals to adaptively navigate their environment, obtain food, and approach or avoid other salient olfactory stimuli. Across all sensory systems, animals use ongoing dynamic sensory feedback to guide targeted movements in space. And in other sensory systems, the key underlying computations for extracting distance and directional information from neural activity are relatively well characterized. But in olfaction, this process is comparatively less well understood. Depending on the nature of the odor source, the environment, and the proximity to the source, animals have access to a number of strategies to efficiently search for and eventually navigate to a target odor source. So in some scenarios, organisms may have clear, unambiguous, and continuously present sensory signals to guide their movement. But in conditions where the cues are more sparse, the search task can become more challenging, and animals may need to work to increase the frequency of detection events for their target odor. Animals may also make use of distance estimation. Olfactory bulb activity grows increasingly intense with odor concentration, and this motivates the prediction that animals could potentially accumulate information about the odor gradient by comparing the relative strength of bulb activity over time. Also, at closer proximity, when there's the possibility of strong spatial concentration gradients, animals may make use of instantaneous comparisons across the left and right nares to obtain a direction estimate. It's clear that odor-guided navigation falters when stereo olfaction is disrupted, but it's not well understood how stereo cues are transformed and interpreted by the olfactory system to inform the animal's search. This is now a rich and very active field of research, and several of our colleagues have carried out refined behavioral work describing specific conditions in which animals appear to be implementing each of these distinct strategies. To take the next step in understanding this complex sensory motor transformation, there's now a real need to ground our models in real-time measurements of sensory activity during motivated behaviors. We're interested in understanding how animals can incorporate olfactory signals as they dynamically update their search strategy and make navigational decisions in a more naturalistic, freely behaving context. To this end, our group, in collaboration with others at BU, has developed a novel head-mounted miniscope design capable of capturing cellular resolution imaging across a 3x4mm field of view. This effectively allows us to image the entire dorsal surface of both the left and right olfactory bulb in animals freely exploring or foraging for odor sources, providing a substantial improvement over the field of view provided by more traditional Grin lens designs. This miniscope weighs 4.5 grams, and animals can move freely and naturally after implantation. And this large field of view could also allow simultaneous imaging of multiple visual or sensory or motor cortical regions in freely moving mice. And we've successfully applied this technology to deeper structures, here imaging activity in large populations of hippocampal neurons through a cannula window. So to observe sensory signals relevant to odor-guided navigation, we applied wide-field miniscopes to image olfactory bulb activity bilaterally during free exploration of fixed odor sources at multiple locations in a 2-foot square area. We used Tibet GCAMP6F mice, which express GCAMP in mitral and tufted cells of the olfactory bulb, and imaged through a thin skull window. The signals we observe here are from olfactory bulb glomeruli. So these flashes arise from a collective neuropil signal that reflects dozens of neurons that represent this sensory channel. The raw signals arising from these recordings are contaminated by fluctuations in the surrounding neuropil and the nearby glomeruli. So we first use an ROI-based non-negative matrix factorization package called FISA to decontaminate the signals for each glomerulus. to the right here is an individual representative example of the signal extracted from one of these glomeruli over time. Now, we ultimately want to link animals' momentary navigational decisions to these neural signals, but the decay of the signal from the calcium sensor is quite long relative to the timescale of behavior. So we therefore further applied deconvolution with some additional smoothing to obtain the signals that we use in the analyses I'll describe today. Here I'm showing you average activity of all ROIs in left and right olfactory bulb during periods when the animal is near two different odor sources. Each odor produces a unique glomerular activation pattern, and these patterns could serve as odor detection or recognition signals to an animal searching for an odor source. Further, as I described previously, if olfactory bulb activation varies reliably with distance from the source, then animals may be able to estimate either the direction of the gradient or the specific location of the source after multiple samples. The intensity and the number of active glomeruli increases with proximity to the stimulus, such that any individual glomerulus has a spatial activity field surrounding its preferred stimulus and produces a distance-dependent tuning curve. If we identify the maximum significant activation distance for each glomerulus, we see the majority of sensitive glomeruli are activated within about 5 centimeters, with a longer tail going out to about 10 centimeters. These data help constrain our thinking about the types of information and the strategies available to the animals at varying distances. First, these data define a minimum detection radius of about 10 centimeters, above which odor detection events will become increasingly sparse. And second, the distance-dependent tuning curves have a general shape where they're steepest closest to the source, and there's an increasing density of active glomeruli at close proximity. So we may expect there to be increased resolution or precision in distance estimation in this range. So animals may further be able to enhance the accuracy and efficiency of their search by making stereo comparisons across hemispheres to determine the direction of the odor source. To support this strategy, glomeruli should show a bias in the directional tuning. I therefore computed directional tuning curves for each glomerulus based on the animal's egocentric bearing relative to the odor source. Here I'm showing a tuning curve for a single example glomerulus computed over periods when the animal is 5 to 10 centimeters away from the stimulus, and we can see little directional preference. However, as the animal moves closer to the stimulus, directional tuning emerges. Now you'll notice that this particular glomerulus is tuned to rightward located sources. Because olfactory input streams are laterally segregated at this stage, the simplest scenario is that rightward tuned glomeruli are in the right hemisphere, and vice versa, leftward tuned glomeruli should be in the left hemisphere. Examining the distribution of directional preferences for glomeruli in either hemisphere, we observe that this is indeed the case, and the strength of this directional bias increases at close proximity. So overall, we can observe odor-specific, distance-dependent, and directionally biased responses in the time-averaged activity of bilateral olfactory bulb glomeruli. Our data indicate that animals may have access to different odor localization information depending on distance. During approach, animals will first obtain detection and distance signals, and then gain directional information very near to the source. This implies that animals may use a serially switching sensory motor strategy at different phases of the approach. Because we're making real-time measurements of sensory activity during free behavior, we can now ask how the dynamic evolution of these signals could inform or influence odor-guided behavior at different stages of odor exploration. So using the average distance tuning curves, I identified a subset of glomeruli that are most sensitive to each odor stimulus. Their activity is shown here in the order that the animal investigates these 3 stimuli in this example video. I'll refer to these as sensors, as a convenient shorthand from now. So rather than smoothly varying with the animal's proximity to the stimulus, these sensors are active in brief intermittent bursts, such that odor detection and distance estimates would appear to be updated during discrete events rather than continuously. To ask whether these signals could support detection and recognition of specific odorants, I tested the accuracy of a naive Bayes classifier trained on glomerular population activity, determining which of the 5 odor stimuli the animal is closest to, and looked at how accuracy varies at different proximity ranges. When using all time samples, performance is relatively poor, but above chance, even at close proximity. I then identified sensor events where the mean activity across the sensitive glomeruli crossed the shuffle threshold. Accuracy is substantially higher using data from these events, emphasizing the intermittent nature of accurate odor information. Now, these signals can clearly provide an estimate of the identity of nearby odor sources. We next wanted to know whether an animal could further extract an estimate of its distance from the source from glomerular activity. I again used a naive Bayes classifier to produce a reconstruction of the movement of the animal towards and away from the stimulus over time, and determine the average error in prediction at different distances. Here, the white trace is the animal's movement, and the blue trace is the prediction using all glomeruli and all time samples. This pink trace is the prediction using only the so-called sensors, and the green here is prediction when training and testing only during these sensory events I described before. The distance estimate can be quite good at very close proximity, and is further improved by focusing in on activity during these sensory events. So this area at which the glomerular distance estimate could conceivably aid in odor localization corresponds to the distances where the majority of sensitive glomeruli are significantly activated. Surprisingly, when we applied the same approach to estimate the odor source direction from glomerular population activity, we're unable to obtain an accurate prediction. In part, we may currently be data limited, and we'll continue to probe this question with larger data sets. However, it's also likely that directional information may be cooperatively computed by pairs of glomeruli in either hemisphere expressing the same odor receptor. Bilateral imaging allows us to identify correlated and similarly tuned glomerular pairs in the same animal. So in future analyses, we can directly test whether making comparisons of the strength or timing of activity in bilateral pairs enhances our ability to predict odor direction. Ultimately, we want to understand how these sensory signals are interpreted and transformed to alter navigational behavior during odor-guided search. To this end, we developed a simple foraging task in which animals must search for sucrose pellets coated with a 20% benzaldehyde solution, distributed randomly and one by one in a dark arena. Here's an example snippet in which the animal appears to pause and collect some sensory information before moving to collect the pellet. To begin to link glomerular activity to navigational behavior, I identified sensory events as before, and examined changes in the animal's behavior around these events. In this example trial, there are 3 events leading up to pellet retrieval. The first one occurs as the animal's nose velocity decreases precipitously. And after the 3rd event, which occurs when the animal is quite close to the stimulus, the animal makes a large change in its trajectory. On average, these sensory events have increasing amplitudes as the animal approaches the stimulus. These events are associated with decreases in the animal's velocity and changes in direction. And these behavioral changes are most robustly observed at closer proximity. So here we have some very strong initial indications that sensory activity modulates behavior on the timescales of individual sensory sampling events. And we're now poised to put this all together to see how animals utilize this information to guide odor localization. Do animals carry out a comparison across sniffs? And do they modulate their movement in response to increasing versus decreasing activity levels? Are they able to make targeted left and right decisions that are related to the directional tuning I showed earlier? And to what extent do they rely on distance versus directional information at each stage of the approach? These are questions we hope to address in the next phase of this research, and I look forward to gathering your insights about our approaches as we move forward. So to conclude, we can now observe the initial sensory signals in both hemispheres of the olfactory bulb during free odor exploration and odor-guided navigation. And these signals are odor-specific, distance-dependent, and directionally biased. During free navigation, odor information arrives in discrete intermittent events, and these events are followed by rapid changes in the animal's navigational behavior. Taking a broader view, These tools provide the opportunity to observe sensory inputs in real time during odor-guided behavior in many contexts. There are clear applications in other navigation conditions where the demands might be more complex, for instance, when navigating in turbulent odor plumes, in natural odor scenes, or when there's background odor noise. And because these are chronic recordings, we can observe the same areas over long time periods. So we'll be able to examine how sensory signals are modulated by learning and plasticity, or the animal's internal state. And so with that, I'll thank you all for your attention. I'll thank the NIDCD and NSF for providing funding, and acknowledge the members of the Davison Lab who supported and provided feedback for this work. I especially want to mention Jiayue Tai, who made major contributions to all the work I described today, and Dan Leman, who pioneered the application of this wide-scope imaging in olfactory bulb. And of course, Ian for providing a great collaborative and productive environment to carry out this work. So thank you.
Thank you, Kevin. Beautiful data. Really, again, fantastic. There are a lot of questions on Slido. I will start immediately because we're a bit behind schedule already. The first one is from Jan Drugovic, and here it is. Are the bursts of olfactory information related to the animal sniff cycles and/or the turbulent nature of odor dispersion in the air?
The short answer is yes, but we can't, almost certainly yes, but we don't have a full view into the answer to that question yet empirically. So we're working to correlate the olfactory bulb imaging signals with respiratory signals, but we don't have that data yet. One proxy that we have, or surrogate that we have, is that I never showed you the raw bulk olfactory bulb imaging data before it's processed, but the background of the image fluctuates in a way that's reminiscent of respiration. So we've been able to extract something that looks a bit like respiration from that data. And if I sort of pretend that's respiration and look for inhalations, etc., then the behavioral changes that I observed with those sensory events appear sort of similar. So I think we're converging on it being a sniff-based input, but also the animal, yes, needs to be near enough odor molecules to have an event.
I see. Thanks. So the second question, I think it's a bit related to one of your last slides, and it was asked before, but let's see if you can provide some more details. So do these mice sniff faster or stronger as they get closer to the source? If yes, is there a way to account for modulation due to sniff changes?
Yes, it's an excellent question, and yeah, I think my previous answer points to it. We don't know the answer yet about the sniffing behavior. We'll be able to sort of, because this is a chronic implant, we can sort of move between a head fixed and a freely moving scenario where we'll be able to get really refined respiration information, but we don't know the answer yet, basically.
I see. So another question from Dorit Derdikman is, how do you differentiate odor proximity and intensity?
Odor proximity is very easy to define empirically. It's the actual distance of the animal from the odor, and intensity, I think, is a much more difficult concept. So, we know that even from our data, it was a bit surprising. We see that even when the animal is very close to the odor, we're not seeing constant, strong glomerular activation. So I think that's one way in which the two are differentiated. The activation only occurs when the animal is close. But otherwise, I think to take a practical or operational view of intensity, I think we would look at the overall activation strength of the glomeruli as a sort of surrogate measure for intensity, and we obviously observe a relationship between the two, but that's one way we'll be able to break those two apart empirically.
I see. So one last one, a question I really like from Victoria Corbett. Were the odors aversive or appetitive? Will the strength of the activation or directional tuning change if the odors were more salient, like signaling reward or threat, for example?
I think it's a really interesting question. We have not addressed that yet. I showed in the last couple slides that we're moving over to a more detailed analysis of the motivated behavior, where the animal really knows that benzaldehyde is an awesome odor that it loves. So we'll be able to focus in on that. I think there might also be, salience or an attentional modulation that might occur, and this gives us the opportunity to ask whether, the animal, when it's seeking out a specific odorant, does any sort of initial filtering to do a better job there. Yeah, we don't know the answer yet, but we're poised to get there now.
I see. Thank you again, Kevin. Sure. It was great having you today.
Thank you.
We're gonna actually move now to the third talk of the session. So yeah, you should leave. Sorry for that. So we're gonna move to the third talk of the session by a talk by Gary Kane. Hi, Gary. How's it going?
Good.
Gary, also from Boston University. And today, Gary, you're gonna tell us about the rate ACC, the anterior cingulate cortex, continuously signals decision variables in a patch foraging task. The stage is yours, Gary.
All right, thanks. And can everybody see slides and everything good?
Yeah, perfect.
All right, cool. Well, I'm Gary, and today I'll be talking about some work in collaboration with John Cohen and Gary Aston-Jones labs, investigating the function of the anterior cingulate, or ACC, during a foraging decision-making task. So generally, foraging refers to the means of obtaining resources, and this can be a very complex problem. So let's look at my favorite forager, Scrat from the Ice Age franchise. His commitment to this acorn should depend on a number of factors. So how long will it take to find another acorn? How long before the ice collapses? So although it may seem stupid for Scrat to risk his life for this acorn, it may actually be a smart decision if it's the only one that he can find. So the ability to make adaptive foraging decisions is critical for evolutionary fitness. Animals that are better at finding food are more likely to survive in the long run. So researchers have been studying optimal foraging theory, the best way for animals to solve these problems, for decades. And to study these questions, theorists have had to place some constraints on the situations that these strategies apply. So I'm going to focus on one model of foraging in particular, foraging in a patchy environment, where resources or rewards are organized in discrete patches and they deplete as they're harvested from the patch. And so animals repeatedly face the decision to stay in the current depleting patch or to search for a new, potentially better one, which comes at the cost of time and/or effort to travel to a new patch. As an example of this, imagine you're eating grapes from a vine. So you first pick off a piece of the vine, and that would be a patch. You find a couple of ripe and juicy ones, but eventually you eat most of these good ones, and it's harder and harder to find them in between all of the disgusting, mushy ones. At some point, you give up, you throw away this vine and pick off another piece. And so that would be akin to traveling to a new patch. The optimal behavior in patch foraging tasks is described by the marginal value theorem, or MVT. And so the goal in foraging tasks is to maximize your global reward rate. You want to get the most amount of reward over time. And so this red line here shows the global reward rate if you decide to leave the patch after X amount of time. So here's the optimal time to leave, where it's at its max. And so now if we look at the local reward rate, or the reward rate in the current patch, you see that this optimal time to leave coincides with the point where this local reward rate dips below this global average reward rate. And so MVT simply points out this simple decision rule that if you leave a patch when the local rate drops below your experienced global or average reward rate, you'll maximize your long-term rewards. So some of the first studies on the neural mechanisms of foraging decisions found some really interesting signals in the ACC. And so historically, the ACC has been associated with cognitive control. And so it's engaged in behaviors that require control, like those requiring focus on a particular stimuli or inhibition of some automatic response. So in foraging tasks, as patches deplete, the need for control increases. And there are a couple of ways to interpret what this need for control may actually mean. So one possibility is that as patches deplete, you need to inhibit this default or automatic response to harvest reward in order to choose to leave the patch. Or maybe as patches deplete, the value of staying in a patch relative to the value of leaving the patch become more similar to one another. So as it depletes, the value of staying is kind of going down and down until you hit this optimal point to leave a patch, which according to MVT is when these values are equal to each other. And so the closer these values are to each other, the more difficult the decision is. And so you may need to exert more control to resolve the conflict from these two increasingly similar options. So regardless of the exact interpretation, the need for control increases as patches deplete, and this is accompanied by increased ACC activity, which has been shown via electrophysiological recordings in monkeys and fMRI studies in humans. And so today I'll present on our efforts to kind of go beyond this notion that ACC exhibits increasing foraging decision-related activity as patches deplete, and to try to better understand what drives the time course of these control signals in the ACC. And this presentation will be focused on a model of rat behavior in an operant chamber-based patch foraging task. So in this task, rats completed a series of trials in which they have to decide to harvest reward from a patch by pressing a lever, or to leave the patch by pressing it or by nose poking in the back of the chamber. So at the start of a trial, cue lights turn on above the lever and in the nose poke, indicating that the rat can now make it execute this decision. And so if the rat chooses to lever press, then there'll be a cue light that turns on in the center reward port, and the rat receives reward as soon as he sticks his head in there. And then there's a short delay, an intertrial interval before the start of the next trial. And so on the next trial, if the rat decides to lever press again, then he can get his reward, but we're going to reduce the volume of the reward to simulate depletion of the patch. And so this happens with every successive lever press. And so after another delay, if on the next trial, the rat decides to nose poke to leave the patch, as soon as he sticks his nose in that nose poke, the box will shut down, so the lever retracts and the cue lights turn off for a 10-second delay, simulating the time to travel to the new patch, after which the lever on the opposite side of the chamber will pop out and the rat can continue to make stay or leave decisions, but with replenished reward volume. So here's a quick video of an example of a rat performing this task. And so it's sped up a little bit, so that's why he looks like the Flash, but he's harvesting from the patch by pressing the lever, and is now considering leaving the patch, and there, just hit the nose poke, and so here's a delay period, and now the rat starts to harvest from the new replenished patch again. And so we tested rats on a variety of patch types that started with a range of reward. So the patches started with as low as 30 microliters of reward and as high as 150 microliters. And they all depleted according to the same exponential decay function. And so we found that rats stayed for more trials in patches that offered greater reward. So you see if in patches that started with 150, they stayed for many more trials than in patches that start with 30 microliters of reward. It's as if they were exploiting these available rewards until they depleted down to some constant leaving threshold. But also, we found that as rats became closer to a patch, so on the x-axis here, this is the trials remaining in the patch, or how many trials until they left. So -6 would be 6 lever presses before the nose poke to leave the patch, 2 lever presses before, and 0 is the trial in which they hit the nose poke. And so across all patch types, we found that their response times were increasing as they got closer and closer to leaving a patch. And so it's well established that controlled behaviors take longer to execute than more automatic ones. And so this increase in response time serves as an indication that rats were actually experiencing a greater need for cognitive control as patches depleted. And so to better understand how rats are making these foraging decisions and to predict neural activity associated with these decisions, we developed an evidence accumulation model of our foraging task. And so this model is similar in concept to a foraging model by Davison and El-Hadi. And there also, I saw a poster yesterday from Sarah Sarosta that used a model with similar computational principles. And so this model estimated the marginal value theorem decision variables on a moment-by-moment basis using leaky accumulators. And so we estimate the stay value, or the local reward rate, by integrating rewards quickly using a strong weight between the reward and the stay value unit. But this value decayed quickly using a very weak kind of recurrent activity. And so you can see kind of the trajectory of the stay value unit during a simulation of the model. And so here, the black line indicates the stay value activity. The dashed vertical blue lines indicate the start of a trial in which the model chose to stay. And the light green dashed vertical lines indicate the start of a trial in which the rat chose to leave. So on trials where the rat chooses to stay, you get this big spike in the stay value as the rat is receiving reward, and then it decays leading into the next trial. And in a patch where the rat chooses to stay for many, many trials in a row, these spikes will get smaller and smaller over time as the reward from the patch is depleting. And so the leave value, we estimated the global reward rate kind of using a weaker weight between the reward and this leave value activity unit, and a stronger recurrent weight, so that you didn't see these big spikes, but it would just get a little bit of a bump with every reward, and it wouldn't decay much. So over time, it basically gave you this constant average reward signal. And so the model makes decisions to stay versus leave using an accumulation to bound process. So we added a leaky competing accumulator layer. And so at the beginning of a trial, when the Q lights turn on, indicating that a decision to stay or leave can now be made, the stay or leave decision units integrated over their respective value units until one of these reached a threshold. And if the stay decision unit crossed a threshold first, then the model would choose to stay on that trial. And so again, on the left, you can see kind of the trajectory of the stay decision and leave decision unit activity over time. And so on decisions to stay, you see a very quick ramp up of the stay decision unit activity. It crosses a threshold, and then it continues to accumulate during a non-decision time delay. So there's this extra delay, which is just allowing time for the model to physically execute the response, the lever press response here. And then after the response is executed, then the activity of this unit will decay back to baseline, leading into the next trial. And vice versa with the leave decision unit activity. When the model chooses to leave, you see this ramp up in activity. It crosses threshold. There's a non-decision time delay waiting for the execution of this nose poke action, and then the activity will begin to decay. So we fit this model to rats' decisions and response times. And we found that it very well explained their decisions, so the number of trials spent in patches and the response times as rats became closer to leaving a patch. So it really captured that rats would stay for more trials when they were given more reward, and it captured that the response times on decisions as they were getting closer to leaving a patch were increasing. And so next, we looked at the activity of these leaky accumulator model units to serve kind of as predicted neural activity. So if ACC encodes these MBT-derived decision variables, such as the value of staying, or that local reward rate, or that global reward rate, then we would expect that ACC activity would correlate strongly with the activity from this model. And so we created peri-event time histograms of each of the model units. And so you can see here the stay value and the leave value, where you get this increase in stay value right after a decision to stay. And the leave value stays relatively constant over time. And so the color of these lines is just indicating the number of trials until leaving. So this would be, again, 5 lever presses, 3, the last lever press before leaving. And this is the trial in which the rat or the model would execute the decision to leave. We also looked at the stay decision and leave decision unit activity, which unsurprisingly on decisions to stay ramps up until the execution of the decision and then decays. And on decisions to leave, you see this inhibition, and then it comes back to baseline, and vice versa with the leave decision unit. And then we also tested 2 additional hypotheses for ACC activity based on prior theories. And so we created peri-event time histograms of a decision difficulty metric. And so this just assumes that as decisions become more difficult, we would expect ACC activity to be greater. And so we took a metric that was just the similarity between the stay value and the leave value units. So the more similar these are, the greater the decision difficulty. And you find that that is increasing as rats are kind of deliberating before making a decision. And then when they're receiving reward, there's inhibition of this decision difficulty, or a reduced decision difficulty. And then we also tested decision unit conflict. So this comes from a long-standing theory of ACC function, which is just multiplicative, kind of the energy between the stay decision and the leave decision units. So when there's pressure, when the activity of both of these is greater, as if there's pressure to both stay and leave at the same time, then you have a greater conflict. But if only one of them is greater and the other is lower, then you don't have as much conflict. So you would still have low conflict even if only one of these values is high. And so we qualitatively compared the peri-event time histograms of recorded ACC activity with these with the peri-event time histograms from the model. And so first we looked at just the average ACC activity. So the average peri-event time histogram across all units, again, locked to the time of decisions. And we found that average ACC activity shared a lot of similarities with the decision difficulty metric. And so both of them were increasing leading up to a decision. We saw a decrease in ACC activity immediately after decisions to stay, but not after decisions to leave. So just only when animals were receiving reward, and then it would continue to ramp up again leading into the next trial. And so, however, this average signal doesn't necessarily indicate that the individual neurons were exhibiting a similar firing pattern. So to investigate the components that went into creating this average signal, what we did is we concatenated peri-event time histograms for each unit across trials in the patch. So we created this matrix where each row represented an individual unit, and then the columns were just the concatenated time points across these 4 peri-event time histograms of 5, 3, 1, or 0 trials remaining in the patch. And then we ran principal components analysis over this so we could pull out kind of what are the components that explain the most variance across all recorded units. And then we can compare the eigenvectors from this resulting principal components analysis. So what were the most dominant components kind of with leaky accumulator model unit activity. And so what we found is that the first eigenvector actually, so the component that explained the most variance didn't look very much like this decision difficulty signal, but it really correlated closely with the value of staying activity. And so both of these are showing kind of decreasing leading up to the decision. There's a spike as they're receiving reward. And across trials in the patch, as an animal gets closer and closer to leaving, it's just decreasing and decreasing. And interestingly, the second eigenvector also didn't look much like this average ACC activity signal, but it did seem to correlate decently with the leave decision unit activity. So on decisions to leave, you see this ramp up, and then kind of right around the time of the decision, it decays back to baseline. And on decisions to stay, it's decreasing, and then after the decision comes back up to baseline again. And so ultimately, we find that this average ACC signal is made up of components that seem to signal some lower level decision variables corresponding to kind of the encoding of foraging value and a foraging decision accumulator. And so we had one final experiment to investigate kind of the contribution of these signals to foraging decisions. And so we pharmacologically inactivated ACC in rats that performed the foraging task. So we implanted cannula targeted toward the CG1 region, and then injected rats with either ACSF as a control or a cocktail of GABA agonists, baclofen and mucimol, right before testing them. And so in these plots, you see red is indicating the control sessions where they received ACSF, and blue is inactivation sessions. And so inactivating the ACC caused rats to stay in patches for a longer period, or for more trials, but it also increased their response times. And so what we wanted to do is we wanted to assess kind of could this be attributed to a change in kind of the value in the perception of value, so in your encoding of local or global reward rate, or is it better attributed to changes in the decision layer, so how we kind of accumulate to kind of come to a commitment to make a decision based on these values? And so to do so, we fit the leaky accumulator model, and then we compared the parameter estimates of fits to the control sessions versus fits to inactivation sessions. And interestingly, what we found is there was only one parameter across all 3 parameters in the model that was significantly different across control and inactivation sessions. And that was a non-decision time parameter, which is not directly part of the decision process. It simply represents the time to execute the lever press or nose poke after committing to stay or leave the patch respectively. And so ultimately, this experiment is kind of helping to elucidate an important limitation of the ACC's contribution to decision-making. So suggesting that ACC's, the signal for a need for control, is not necessary to stick to an established decision strategy or to make a decision based off of a stable internal model. So in foraging tasks, no learning is required because the animals should already have kind of a representation of the distribution of possible outcomes. And so in this paradigm, we don't see that the ACC is kind of playing a critical role in following the important decision strategy, kind of sticking to the marginal value theorem per se. And this is consistent with a recent study published using optogenetic inactivation of ACC in mice performing a foraging style task. However, the ACC seems to be very important in tasks that require updating internal models. So if there's a change in the environment or a change in the rules of the task, in these kinds of paradigms, inactivation of the ACC does affect kind of animals' ability to update behavioral strategies. So it seems like maybe this is where this control signal comes in more handy, and not necessarily for adherence to a constant strategy. Okay, and so as an example of this, you can see kind of Akam and colleagues from a paper from 2020. And so finally, I'd just like to thank my PhD advisors, John and Gary, for their support throughout this project, and my current mentor at BU, Ben Scott, as well as other collaborators, Amitai, Bot, Nathaniel, and Morgan. I'd also like to thank our funding sources, and thank you all for your attention and for the organizers for putting together such a great conference.
Thank you, Gary. Really cool work. Very, very interesting. There are already several questions. We're a bit late, so we'll take perhaps just a couple. The first question is from Jason Moore. How much animal-to-animal variability do you observe in rat strategies? Does animal age correlate with fitted model parameters, for example?
I can't speak to age. That's something that I've never directly looked at. I would say that, I mean, 100% of the rats that I have tested follow this general strategy. There is individual variation. So some rats will stay longer in patches and some rats will tend to leave earlier, but they all follow kind of these general predictions.
No, I see. Thanks. So the next question is from Roxana Zerati. How do you guess expect the neuronal activity and behavioral change if we have food patches without sharp boundaries, instead having smooth distributions?
That's a very good question. I think ultimately it's going to depend a lot on what the rat can learn about the environment. So I think if the rat can make really good predictions and has a good idea of when this patch is going to end. So in other words, if it can keep these prediction errors low, despite having these sharp changes, then I think a similar model, like this kind of model, should still be able to predict what's going on. I mean, that is if it's an accurate model, but that would be a great way to sort of invalidate it. So I guess I would expect that maybe if it was very predictable when that sharp decline was going to be, then maybe decisions wouldn't become more difficult over time gradually. And so you might see kind of sharper, quicker changes in activity or something like that. I think that would be my best guess.
Mm-hmm. Great, thanks. Yeah, interesting. Like last question, sorry for the other people who ask a question. Next question from Linda Yu. Have you recorded from any other prefrontal areas in addition to the cingulate cortex? And if so, did you find any similar decision variable there?
Have not recorded from other prefrontal areas, so can't speak to it, but I think those would be very interesting experiments.
Can you speculate, perhaps?
I mean, I think based on prior literature, then I would expect to see kind of some reward signals, maybe like more activity related to like pressure to like stay in a patch from say like orbital frontal. I think that's one of the regions that has been like very kind of more well studied in foraging tasks, but I'm a little bit scared to speculate too much.
Don't be too scared. All right, thank you, Gary. Thank you very much.
Yeah, sorry. Just lastly, if somebody wants to send an email, I know I went a little over to, I'm happy to discuss further. So gakane@bu.edu. But
or on Gazer Town later.
Oh yeah.
Thank you, Gary. Bye. All right, so now the last talk of this session, and I guess we're shifting animal species here. So the next talk will be by Jenny Liu. Hi, Jenny. I hope everything is doing all right. Jenny, you're from Harvard Medical School, and today you're going to tell us about transformation of velocity signals from egocentric to allocentric coordinates. Jenny, please go ahead. Yes, perfectly.
So thank you, Adrien, and thanks to Leslie and the organizers for giving me the opportunity to present our work. In order to navigate, animals need to know how they're moving through the world. So these movement signals are typically relayed in the brain as body-centric or egocentric coordinates. So for example, moving left, right, forwards, or backwards. But these egocentric movements can mean different things when referenced to the external world. So for example, you could be heading north and walking straight forward, or heading west while sideways walking to the right, or heading south but walking backwards. At the end of the day, you're traveling north. And so this travel vector is therefore in a world-centric or allocentric reference frame. Like us, insects aren't always moving straight forward. Flies can walk backwards and make significant lateral movements while turning. So they, like us, need to relate their body movements into world-centric coordinates. In order to perform this transformation, the brain needs to have two key pieces of information. It needs to have heading or compass cues, and it needs to have velocity cues, which is a vector with magnitude and direction. In insects, we know that heading direction is encoded by compass neurons in a toroidal or donut-shaped region called the ellipsoid body. Compass neurons are in the donut are arranged topographically, as shown here, according to their preferred heading direction. Compass neurons, also known as EPG neurons, make topographically arranged projections to the protocerebral bridge, or PB, forming two complete maps of heading, one on the left and one on the right. In this project, we wanted to understand how walking velocity signals might be combined with compass heading signals. And so we decided to start by looking at cell types downstream of the compass neurons in the PB. We're using 2-photon calcium imaging to look at the population activity of a cell type. These flies walk on a ball tracked in real time using machine vision software, and they're in a virtual reality display with a heading landmark. So if the fly rotates left, the VR world moves right. So I'm going to give you a roadmap, and I'm going to start with our conclusions. So I'm going to tell you today about a circuit in the fly that performs an egocentric to allocentric transformation of velocity. This circuit consists of two steps. So in the first step, EPG neurons, which encode heading, synapse onto two cell types called PFND and PFNV. These PFN neurons conjunctively encode heading direction and the egocentric body velocity. In the next step, PFN neurons project to a structure called the fan-shaped body, where they converge onto a cell type called H delta B. And this step computes an allocentric travel velocity. So now I'm going to walk through the circuit, starting with the first step. I'll start by showing the population calcium activity of EPG neurons in the PB, which, if you recall, have two complete maps of heading. So time is increasing down the y-axis, and each row represents the calcium activity along the entire length of the PB. And you can see here that there are these two spatially localized bumps of calcium activity. We can plot the position of the bumps in circular coordinates here in gold, and you can see that the bump position aligns with the position of the VRQ. So EPG neurons encode the heading of the fly. The two bumps are symmetric, regardless of the fly's velocity, which I've shown here on the right panels. So now I'm going to show you that calcium activity of PFND neurons in the same format as I've shown here. PFND neurons also have two bumps of calcium activity, and the bump position also tracks the VRQ position. So PFND neurons encode heading. What's different is that the bumps are changing in amplitude and they're not always symmetric. So if you pay attention to the part that I've highlighted here, you can see that the fly is walking forward and to the left, and the left PB bump is far brighter than the right. We can plot each bump's amplitude as a function of forward and lateral velocity. So here the bump responds to forward and ipsilateral walking. So the right bump grows when the fly walks in the forward right direction and vice versa. And the bump grows with increasing speed along that direction. We can represent this as a vector showing the preferred egocentric velocity direction for each bump. So the right bump responds to forward right walking, and the left bump responds to forward left walking. PFNV neurons also have two heading bumps in the PB. So here the bump amplitude responds to walking in different directions. So for example, in this specific moment, the fly walks in a backwards left direction, and the right bump amplitude increases. Across flies, we see that the amplitude of each PFNV bump increases with backwards contralateral movement. So if we represent this as vectors, the right bump grows with backwards left walking and vice versa. So in summary, PFN neurons conjunctively encode the heading and egocentric translational velocity of the fly. PFN populations do this by having a spatially localized activity bump whose phase encodes the heading of the fly and whose amplitude encodes egocentric velocity. When we overlay the preferred velocity directions for each of the 4 PFN subpopulations, we can see that they roughly create a Cartesian system. So in other words, the amplitude of the 4 PFN calcium bumps can be used to encode translation velocity in any direction. This Cartesian system is in an egocentric reference frame. So when the fly rotates in space, these vectors rotate along with it. Now, I'd like to revisit our original goal, which is to convert egocentric velocity into allocentric coordinates. And remember that these 3 flies have different combinations of heading and egocentric velocity, but they're all traveling in the same world-centric direction. So PFN neurons are encoding these different combinations of heading and egocentric velocity. And so downstream, the circuit needs to figure out how to make these different combinations equivalent. Well, it turns out from connectomics data that PFND and PFNV neurons converge onto a cell type called H delta B. And the key to this coordinate transformation is in the wiring. I'm going to introduce this anatomy in the next few slides. So here I'm showing you a simplified anatomy schematic for PFND neurons, where the dendrites are the filled squares and axons are open circles. If we just look at the neurons from the left PB, we can see that they make topographic projections across the entire width of the fan-shaped body. And the same happens for the right population as well. PFNV neurons have the same projection pattern, so just know that the same diagram works for the blue neurons. H delta B neurons have both dendrites and axons in the FB, separated by approximately half the width. So if the FB represents 360 degrees, then H delta B neurons are connecting 180 degrees apart. So now I'm going to break down the wiring of PFN onto H delta B neurons into 2 key features. The first feature is how H delta B neurons combine opponent inputs. So here I'm showing you one H delta B neuron in purple, and I'm also showing you the major inputs to this H delta B neuron from the left PB, where the red PFND neuron actually targets the axon, and the blue PFNV neuron targets the dendrite. The black arrows on top are the preferred heading directions for the PFN neurons. So you can see that this H delta B neuron is hearing from the red and blue neurons that have preferred heading directions approximately 180 degrees apart. And the same is true for the inputs from the right side. So now I'd like to remind you of our Cartesian system of preferred egocentric velocity. You can see that the right PFN populations encode opponent directions in egocentric space. And the same is true for the left PFN populations. So what this subcellular targeting is doing is making egocentric translation in opposite directions equivalent to changing your heading direction by 180 degrees. You can also think of this as a rotation of these opponent vectors so that they constructively combine downstream. The second feature that we notice is the wiring of PFN neurons between hemispheres. So here I'm showing you one H delta B neuron in purple and the major inputs from the red PFND neurons from the left and right side. Now, if we look at their preferred heading directions, we can see that there's a difference of approximately 45 degrees. Now we can bring back the preferred egocentric velocity vectors, and we can see that they're separated by a similar amount. So again, the angular difference in preferred heading is equal and opposite to the angular difference in preferred egocentric direction. Put another way, this wiring rotates the left and right egocentric velocity vectors so that they constructively combine. And the same story applies for the blue PFNV neurons. So let's bring this all together. I'm going to show you one H delta B neuron and our 4 major input neurons. Now I'm also going to bring in our preferred egocentric velocity vectors. Let's see what each input PFN neuron encodes in world-centric space. So for the left PFND neuron, we can take our fly and align her heading to the preferred heading direction. Then we keep the vector corresponding to the left PFND's preferred egocentric velocity. And so this arrow now represents the fly's velocity in allocentric space. And then we could do the same thing for all 4 input neurons. And we find that all 4 flies are traveling in the same general direction. So in this case, northeast to east. Now, these travel directions aren't perfectly aligned. And the reason for this is because the schematic I'm showing you here is actually pretty heavily simplified. But it should give you some intuition that the PFN to H delta B wiring is integrating egocentric velocity and heading direction combinations that are equivalent in allocentric space. We wanted to formally test these insights using connectomic data and our physiology data, and we collaborated with Lydia Hamburg and Shaul Druckmann at Stanford to build a computational model of this transformation. This model takes in the heading of the fly and the translational velocity of the fly, shown in green. The translational velocity is projected along each PFN neuron's preferred velocity vector, and that value is rectified to generate a magnitude. This magnitude is used to scale the amplitude of the heading tuning of a given PFN neuron, for which we use a cosyne function. And then finally, H delta B neurons simply sum the inputs from PFN neurons. And importantly, we take connectivity directly from the Janelia heavy brain connectome, and the weights of those connections are simply proportional to the synapse counts. The output of our model showed that there was also an activity bump in the H delta B population. So we could look at the bump position by taking the population vector average, which is displayed here on the y-axis in circular coordinates. On the x-axis is the world-centric direction of the fly. And each line is showing this relationship for different heading directions of the fly. And you can see that the model predicts that the H delta B bump position linearly encodes the allocentric travel direction. And this is invariant to heading. We could also introduce perturbations to our model. So for example, if we remove PFND or PFNV neurons, we find that the position of the H delta B bump basically reverts to encoding the heading direction of the fly. So if a fly is walking straight forwards, its travel direction is aligned with heading. But if a fly is walking backwards, the travel direction is opposite from the heading. So we designed an experiment to look at H delta B activity when the fly walks backwards. So here the fly is in VR with a landmark cue, and every minute we show a looming stimulus in front of the fly. Here I'm showing you data from one experiment. On top is the radius of the loom, and below is the fly's velocity. So you can see as the loom reaches its maximum radius, the fly makes a large backwards movement. Here's a population calcium activity of H delta B neurons, where each column shows the delta F over F across the entire width of the FB. You can see that there is a spatially localized bump. Now we can plot this bump position in circular coordinates and overlay the VR landmark position. At the beginning of the trial, the fly is walking fairly straightforward, and you can see that the VRQ position and bump position align fairly well. But when she makes this backwards movement here, there's a large deviation of the bump position, which is what we expect if H delta B neurons are encoding travel velocity. We've seen this effect now across multiple trials in a number of flies. So in summary, I've described to you a circuit in the fly that transforms velocity from egocentric to allocentric coordinates. This computation would allow the fly to relate its movement to external cues and goals, and we think it might be critical for behaviors such as path integration. I think this circuit is a great example of how wiring architecture can accomplish complex computations like coordinate transforms. And it also illustrates the power of combining physiology with connectomics for circuit analysis at a fine resolution. I'd like to thank my advisor, Rachel Wilson, and my colleagues and collaborators who made this project possible, including Elena Westeinde and Paul Dawson from the Wilson Lab, and Lydia Hamburg and Shaul Druckmann from Stanford. I also like to thank Cheng Liu and Gabby Mayman for providing us with the H delta B split line. And I'd like to mention that they have a parallel study, which you could read in addition to ours on bioRxiv. And thanks to all of you for listening.
Thank you, Jenny. Wonderful talk, beautiful data. Please ask your question on Slido for this last talk. We already have a first question from Ilya Nemanman. And I will actually rephrase it a bit, I guess. So does this compass is this compass fully driven by vision, or do you think that there are other sensory inputs here that are at play, like auditory, or perhaps actually even some kind of proprioception for the movement direction?
That's a great question. We know that various insects, for example, dung beetles, use visual cues as well as wind cues for determining their compass heading. And work from our lab has shown that wind cues can enter the central complex, namely the ellipsoid body, where they could potentially influence heading direction of the fly.
I see. Thanks. So Dory Druckmann now is asking, like, I guess the billion-dollar questions, which is that whether is there evidence for position coding downstream of these neurons, given this nice path integration allocentric velocity signal that you've shown today?
We think there might be, but this is we're really interested in seeing how these signals end up getting integrated downstream. Yeah.
I see. Okay, cool. So, but any sign, like, how does it guide behavior? Basically, like, what do these neurons do?
Oh, I guess okay, I understand your question. So, I think it's fairly important to be able to know, to integrate backwards walking movement and lateral movements. So flies walk backwards, and when they turn, they make a lot of lateral movements. So it could be interesting and important to see whether it's involved in distance estimation or path integration. And we know that flies can have path integration behaviors. So for example, they will keep track of the location of a food source.
I see. So there's another very interesting question from Alexei. Is the complex wiring that you've described today, do you think it's acquired, or is it all hard-coded in a genetic code?
That's a great question. So in Drosophila, cell types, there tends to be consistency in cell types in the brain. So there's, it's unlikely that, for example, this wiring is learned or entirely driven by plasticity mechanisms. But it'd be interesting to see whether there's learning and plasticity in these central circuits.
I see. Thank you very much, Jenny. I think that will conclude this session. Thank you to the 4 speakers. Thank you to everyone who participated in the discussion by asking questions. Thank you again to the organizers for putting this great virtual conference together. I hope you're enjoying it as much as I am. And yes, so this is the end of the session. Next session will be a bit later, actually in 1 hour from now. And have a great time, everyone. Bye.